Remote control for plants. Plants have infinitesimally little pores on the outside of their leaves called stomata. These assist plants with directing the flood of carbon dioxide for photosynthesis. They likewise forestall the deficiency of a lot of water and shrinking away during dry season. The stomatal pores are encircled by two gatekeeper cells. In the event that the inward pressing factor of these cells drops, they loosen and close the pore. In the event that the pressing factor rises, the cells move separated and the pore broadens. The stomatal developments are subsequently managed by the Watchman cells. Flagging pathways in these cells are mind-boggling to the point that it is hard for people to intercede with them straightforwardly. Notwithstanding, scientists of the Julius Maximilians Universität, JMU, Würzburg in Bavaria, Germany, all things considered figured out how to control the developments of stomata distantly, utilizing light heartbeats. Light touchy protein from green growth utilized. The scientists prevailed with regards to doing this by bringing a light delicate switch into the gatekeeper cells of tobacco plants. This innovation was received from optogenetic. It has been effectively abused in creature cells, yet the application in plant cells it is as yet in its earliest stages. The group drove by JMU biophysicist and Watchman cell master Professor Rainer Hedrick portrays their methodology in the diary Science Advances. JMU specialists Xiaoguang Huang, first creator, Kai Conrad and Rob Rolfsema were fundamentally included. The gathering utilized a light delicate protein from the alga Gaillardia theta as a light switch, in particular the anion channel ACR1 from the gathering of channel rhodopsins. Because of light heartbeats, the switch guarantees that chloride streams out of the Watchman cells and potassium follows. The Watchman cells lose interior pressing factor, loosen and the pore closes inside 15 minutes. The light heartbeat resembles a controller for the development of the stomata, says Hedrick. Anion channel theory affirmed. By presenting ACR1 to light, we have crossed over the phone's own flagging chain, in this way demonstrating the theory that the kickoff of anion channels is fundamental and adequate for stomatal conclusion, Hedrick says. The openness to light had totally forestalled the happening of the plants. With this information, it is presently conceivable to develop plants with an expanded number of anion directs in the gatekeeper cells. Plants prepared in this manner should close their stomata all the more rapidly because of moving toward heat waves and consequently be better ready to adapt to times of dry season. Plant anion channels are initiated during pressure. This cycle is reliant upon calcium. In a follow-up optogenetic project, we need to utilize calcium-leading channel rhodopsins to explicitly permit calcium to stream into the Watchman cell through openness to light and to comprehend the instrument of anion divert enactment exhaustively, Hedrick says. Fundamental logical examination can likewise profit with the outcomes from Würzburg. Our new optogenetic instrument has tremendous potential for research, says the JMU teacher. With it, we can acquire new bits of knowledge into how plants control their water utilization and how carbon dioxide abscission and stomatal developments are coupled.